Hello dear students, this is Lázaro Pino, your chemistry professor, and today we are having lipids, classification and application to health sciences. We can say in general that they are biological compounds non-soluble in water but in non-polar solvents. They are very heterogeneous. They can be saponifiable. It means that they can be hydrolyzed under basic conditions or non-saponifiable. Also, they can be divided into simple and complex ones. The structure and classification of the lipids is in 5 categories. 1. Energy storage lipid, membrane lipids, emulsification lipids, messenger lipids, and protective coating lipids. Here we have fatty acids. Fatty acids are small units that make more complex fat or lipids. The fatty acids are divided into a long non-polar chain and a polar head, that is the carboxylic acid. Fatty acid could be long chain from 12 to 26 atoms of carbon, medium chain from 8 to 10, and short chain when the number of carbon is no more than 6 and as much as 4. The unsaturated fatty acids, as you can see here, present one or more unsaturations or double bonds. The notation for fatty acids that are saturated is, for example, 16 to 0. It means that it has 16 atoms of carbon and 0 double bonds. When you have this notation 18 to 1, delta 9 means that it has 18 atoms of carbon, 1 double bond that is in carbon 9, and so on, as you can see here. Here you can see different fatty acids. The saturated ones that had atoms of carbon from 14, 16, 18, 20 has boiling points of 54, 63, 70, and 78. What happened with them? That they are in the solid state at room temperature. Then we can say that saturated fatty acids are at room temperature solids, while the monounsaturated and polyunsaturated, as you can see here, at room temperature, they are liquids because the melting point is too low. As the number of atoms of carbon increase in saturated fatty acid, the melting point increases too. But for the same number of atoms of carbon in unsaturated fatty acid, we can see that when the number of insaturation increase, the melting point lower. Which one are the properties of fats and oils? Well, triglycerol in natural fats and oil are non-polar. They are hydrophobic molecules with no ionic charges. Oil is a mixture of triglycerol that is liquid because it contains high proportion of unsaturated fatty acid, and fat, a mixture of triglycerols that is a solid because it contains a high proportion of saturated fatty acid. Here you can see omega-3 and omega-6 that are important fatty acid used as nutrition supplements. The In the group of energy storage lipids, we have the triglycerides. The older term was triglycerols. They are made of a glycerol that is a polyalcohol and 3 fatty acids that are joined making esters. The triglycerol then are lipids formed by esterification of 3 fatty acids and a glycerol molecule. Both fats and oils are complex mixture in which many different kinds of triglycerol are present. Generally, fats are obtained from animal sources, while oils are obtained from plants. Fats involve triglycerols that contain many saturated fatty acid components, while oil have more unsaturated fatty acid components. Here you can see in this graph, for example, canola oil that contains only 6% of saturated fatty acid, 58% of monounsaturated, and 36% of polyunsaturated. While coconut oil contains 92% of saturated fatty acid and only 6% of monounsaturated fatty acid. What is a wax? Wax are made of a long chain fatty acid with a long chain of alcohol. Here we have, for example, methyl palmitate that has a fatty acid and a long chain of an alcohol. Phosphoglycerol are those ones where you change one of the fatty acid by a phosphate group and an alcohol group. Here we have phosphatidic acid. The alcohol that are in the phosphoglycerides are, for example, choline, serine, and ethanolamine. Phosphatidylcholine, in other words, lecithin, is the one that is presented here. The lecithin is used as a emulsifier. Dipalmitol phosphatidylcholine formed as a surfactant in lung cells. Here we have a cephalin found in cell membranes. Sphingolipids are made of sphingosine plus a fatty acid plus something else. Here we have an example of it. The general configuration of sphingomyelin is the backbone is sphingosine, a fatty acid, a phosphoric acid with choline. These sphingomyelins are found in myelin sit around the neurons. Sphingomyelin is, as I told you before, is made of a fatty acid, a phosphate group, and choline. Here you have another one where you have a carbohydrate too. Here you can see the carbohydrate. Different diseases that can be because of lipid storage are this one that you can see here in the brain, in the liver, and in also in several particular liver and spleen. Here are the diseases and the type of lipid that is accumulated in each case. The biological membranes separate the cell interior from exterior of cellular organelles from the cytoplasm. About 60% lipid and 40% of protein is in them. And we found in the cell membrane the phospholipid bilayer. As you can see here, the lipid bilayer or phospholipid bilayer is made of non-polar tails and a polar head. Here you can see the double structure of a cellular membrane. And here you have an image of how it is composed. Also we have cholesterol, we have carbohydrates, but mainly it's a lipid bilayer, a phospholipid bilayer. Here you can see another important of lipids, blood type antigen. Here we have the 0, the A, and the B antigen. They are made of, as you can see, glucose, galactose, and fructose units. And they are joined to sphingosine and fatty acid units. Steroids are non-saponifiable lipids that comes from cholesterol. In order to understand the structure of anyone or recognize it, you have to see the 3 hexagons and 1 pentagon that are together. This is the skeleton of cholesterol and all the steroids. Here you have the cholesterol structure. It is found in the diet of animals, and also is synthesized in the liver. Cholesterol is an essential component of the cell membranes. It's used as a raw material for sex hormones, and also make bile salts. High blood levels are associated with heart disease like atherosclerosis and arterial plaques. Here you can see how cholesterol is transported as a lipoprotein complex, the LDL. LDL is high density lipoprotein. LDL is low density lipoprotein. VLDV is a very low density lipoprotein, and we have also chylomicron, mostly triglycerides. In this graph, you can see the biological flow of cholesterol and how you can find the then in different paths during the metabolism in our body. Bile salts act as emulsifying agents to digest fats, and they can produce a stone when cholesterol gets too concentrated. Also, we can find adrenocorticoid hormones. Here you have some example. Glucocorticoids regulate blood glucose levels, cortisol and cortisone. Also regulate inflammation and are used in treat inflammatory diseases. Mineral corticoids that has influence in the level of sodium and chloride in blood, and aldosterone that is very, very important. Here you have the structure of them. As you can see, all of them, the skeleton of them is the same skeleton of cholesterol. That's why they are called steroids. Male hormones, male sex hormones are testosterone. They are produced in the testes, and they produce the secondary sex characteristic, the muscle growth, and so on. But sometimes it is used in a bad way by athletes and other type of men, because they have bad side effects. Female sex hormones are estrogen, like estradiol and estrone, and progesterone, and they are produced by the ovaries. And they are very important in the reproductive cycle and produce a secondary female characteristic. Hormones like the prostaglandins are very important to regulate fever, contraction, inflammation, induces the labor, and asthma, made from arachidonic acid. Then, from the arachidonic acid, you can produce the prostaglandin. It's a natural mechanism. Then, the classification of lipids that we have studied today are those ones that are saponifiable lipids and the non-saponifiable lipids. In the saponifiable lipids, we have simple and complex ones. Simple waxes, triglycerides, phosphoglycerides, and sphingolipids. And among the non-saponifiable lipids are steroids and prostaglandins. Well, that's all for today. I hope you understand it, and if not, try to go over this several times. These structures are very important. They will be in the final exam.